Hi, welcome to Lab Techniques in Biology. This video is being made specifically for students prepping for the Medical College Admission Test, or MCAT, but I also hope it helps any biology student who feels like they have a difficult time understanding biology problems because they don't understand how the lab or research components in those problems work. So we're going to be going over a wide variety of techniques and how they work, and also how they might show up in your MCAT passages on the biological and biochemical foundations of living systems section, and a little bit from the ChemPhys section as well. I hope to get through this in one part, but it may turn into a part two, because I love research. My experience in the two labs that I worked in biochemistry and cell biology, greatly helped me understand passages, so any knowledge I can impart about how this works, I hope it will do for you what it has done for me. So starting at the beginning, a lot of passages you will come across, and a lot of just biology experiments that you may be told about in general, involve cell culture. So this is where we take a tissue culture dish and we grow cells in vitro, or not part of a living organism, but those cells are alive. So they will form a tissue, aka cells that are all the same and all perform the same function. So for example, in one lab I worked with human embryonic kidney cells, or HEK cells, which is a common lineage to use. And basically, because they were embryonic cells, they could go on forever and ever. Another line that I worked with was called human osteosarcoma, and I know that sounds like a cancer because it ends in like oma. And it was a cancer cell line, and this is used a lot in biology because cancer cells, as you might know, proliferate like endlessly. That's their problem in living organisms, but they're very useful to us in the lab. One famous cancer cell line you might have heard of is the HeLa cell line. This cell line came from Henrietta Lacks, and the story is a bit unfortunate because she was a black woman who at the time, medicine was not kind to black women. It still is not, but she did not consent to her cells being used, but they are used very commonly and ubiquitously today. But the point is that cancer cells are useful to us in the lab because they proliferate so often, and they're basically immortal. So it's cancer cells and embryonic cells you'll see often. Usually, if you're on the MCAT, the passage will tell you everything that you need to know about the cell line, but basically know that cell culture is where you can take cells and keep on seeding them in new dishes and keep them going indefinitely if you do it right. So now we're going to turn our focus to centrifugation. This is another important technique in biological labs. So basically the way that this works is you'll have tubes that balance each other out in a spinning apparatus called a centrifuge, and there's going to be a force from the circular motion that basically forces everything down in the tube. You can have something called a density gradient, and we use this to measure various things, to filter things. You might have heard of ribosome densities that are different in prokaryotes and eukaryotes, and that was measured with a density gradient, where at the top of the tube you have really dilute solution, and in the bottom of the tube you have really concentrated solution. And as you go down, the difference between the centrifugal force and the buoyant force changes, and then you'll get different densities of things you're looking at with the different densities in the tube. Another thing that centrifugation is used for is like cell fractionation, where if you have whole cells and you lyse them, then you can centrifuge, and all the heavy things in centrifugation go to the bottom. So like your mitochondria and your nucleus and stuff like that will go to the bottom, and everything in the cytosol will stay in what we call the supernatant, which is the liquid that goes on the top of the tube, and that is not part of the pellet that forms beneath. So you can easily pour off the supernatant and leave yourself with the pellet, and then resuspend or mix up the pellet with new solvent. So this is a very useful technique, and one that you'll definitely see around. This is just another slide showing how this works. And if you are my student and you have my contact information, you're welcome to ask me for these slides. I do have them. So now I want to look at mass spectrometry, and this is something you might see more often in chemistry. You might have learned it in organic chemistry, and it does show up in biology sometimes because we like to mass spec proteins and other biological molecules in order to identify them. Mass spec works in that you actually shoot a beam of electrons at whatever molecule that you're working with, and you will create several different cations or positive ions because the electron beam is going to knock off other electrons that either are part of the molecule as a whole or form bonds. Now these fragments that we make, these cation fragments, are going to reach this detector that we're using at different speeds. And this is based on something called the mass to charge, or M/Z ratio. Usually in mass spec, I've never seen it different than this actually, the charge is +1. So the mass to charge ratio is really a fancy way of us saying the mass. So the mass spectrum machine contains a magnetic field, and that magnetic field is going to curve the paths of things by different amounts, and that's dependent on the mass. Because if you think about it in a physics sense, and you can tune me out if you haven't done this part of physics, force is mass times acceleration. So if you have a magnetic force going on these particles, on these fragments, then if they have a bigger mass, they're not going to go as fast. So you're going to end up with little things hitting the detector first and bigger things hitting the detector later. And in the MCAT, they love to use mass spec and similar techniques like Maldian passages. And what they'll do is they'll basically apply this concept of these particles with a positive charge accelerating toward a detector to the physics of electric forces and electric potential energy. So basically, if you get a passage with mass spec and then it's asking you physics questions, try to think of it separately a little bit. Try to just focus on the physics and think of a charged particle accelerating through an electric field and what you know about that. So now I want to look at something called electrophoresis. So SDS Page, what's on this slide is a specific type of electrophoresis, and then we'll see this again. But the concept of electrophoresis is that if you have a molecule, and in most cases it's a biological molecule, and you put it at one end of what we call a gel, which is just basically a material that has a lot of pores or little holes in it. And this gel gets charged with a voltage such that one end is negative and one end is positive. If this thing has a certain charge, we can make it be attracted to the other end of the gel, and then it moves through the gel. One of my favorite biochemistry professors told me this analogy about protein gel electrophoresis, that think of a chain link fence. Like if you've ever been to a park with a baseball field, there's one near my house and it's got a chain link fence with like little holes in it. Imagine throwing a baseball at that. It's probably not going to go through, right? Now imagine throwing a golf ball at it. Easier to get through, right? So that's the thing with proteins in SDS Page, which Page, by the way, stands for polyacrylamide, which is the gel material, gel electrophoresis. We'll talk about the SDS in a minute. But in this electrophoresis, we've got proteins of varying sizes trying to get through these pores in this gel. So this electrophoresis will separate things out by size because smaller things can get through more pores, and it basically gets farther down the gel in a given amount of time. As you might have guessed, we're also separating things by charges. If something is more highly charged in SDS Page, it's going to be more attracted to the other end of the gel. That's going to help it travel farther because it's got a bigger force of attraction, which leads to a bigger acceleration if you assume the two things have the same mass. So in gel electrophoresis of any kind, we're separating things by mass and by charge. Now, what is the meaning of this whole SDS thing? Well, in proteins, remember that we have various levels of structure that build up our protein. So we have primary structure, which is our sequence, we have secondary structure, which is alpha helices and beta sheets, and we have tertiary structure and quaternary structure, which are the overall 3D structure of the protein. And in the case of quaternary, it's the protein interacting non-covalently with other subunits, right? So we have a concept that goes with that, and it's called denaturation. And denaturation basically means that all of those non-covalent interactions, so everything except for primary structure, is being broken. Now, SDS is a compound that does that. It's a denaturant. So if you do SDS Page, you're going to denature your proteins. So that being said, if you do have a protein like hemoglobin that is multiple subunits, you're going to be breaking up those subunits and treating each protein as a monomer. And that's going to matter if they give you weights, which will often be seen in kilodaltons. A dalton is a measure of molecular weight, and then kilo is just our prefix, right? So you're going to see that application, like if you have a tetramer, which is a 4 subunit protein, and the tetramer is I don't know, 10,000 kilodaltons. I'm bad with these numbers, so that may be ridiculous. Apologies in advance. So then you would divide 10,000 by 4, and you would get like 2,500 for each subunit. And that's the number that you would expect to see in your SDS page. And usually in gel electrophoresis, you'll have a ladder, which is like a control group that has molecules of certain masses so that you can see where they're supposed to end up on the gel. So why does it matter so much that proteins are being denatured? Well, sometimes you don't want to denature them. Like, what if I want to tell if something is a monomer or if it's a dimer or trimer or tetramer? I want to keep it intact. That is something called native page. So you're going to keep the thing in its native or its natural conformation or shape. So in native page, you could tell the difference between a protein that's a monomer and a protein that's multiple subunits, because the multiple subunits would stick together and then they wouldn't get as far in the gel because it's bigger. Another thing to consider with page is that think about the majority of tertiary structure and secondary structure interactions are non-covalent, right? When we denature things, we break non-covalent bonds. We do not break covalent bonds. The exception to this entire like tertiary structure as non-covalent thing is disulfide bridges. So disulfide bridges are where we have two amino acids called cysteine, and they each have a sulfhydryl or thiol group. And this starts out looking like this. So you're going to have two thiol groups. And we're going to oxidize it. So this process involves oxidation. And we're going to make a disulfide bridge. So some proteins have this. It's a disulfide bridge. So you can have different monomers connected in like a trimer or a tetramer, but they're not connected via non-covalent interactions. They can be connected via disulfide bonds or disulfide bridges, which are covalent bonds. In this situation, if you denature this protein of multiple subunits with SDS, it's going to stay in its polymeric form. It's not going to break into monomers because you don't break covalent bonds. This is where reducing and non-reducing conditions come in. So if you have a reducing protein electrophoresis, that means that you break disulfide bridges. If you have a non-reducing protein electrophoresis, you are not breaking disulfide bridges. So those are the things to keep an eye out for. We have SDS page, which denatures. We have native page, which doesn't. And either of those can be reducing or non-reducing, which means A, you break disulfide bonds, or B, you do not. So this is a super useful technique for telling us how much protein that we have. And often this is worked up in something called a Western blot. Now, Western blot kind of has a funny story for how the name was made up. It's not from the West. There's another technique we'll cover later on called Southern blot, and this has to do with electrophoresing DNA. Now, there was actually a guy called Southern. That was his last name. And people in biology, when they started to electrophorese other things besides DNA, like RNA and protein, decided to run with the compass theme. So I like to draw the little compass and say that Southern blot is DNA, Western blot is protein, Northern blot is RNA, and Eastern blot actually doesn't exist. And all we mean when we say blot is we do a workup that transfers the things on the gel onto a different surface so that we can image it. All right, and then we have isoelectric focusing, which is a specific type of protein electrophoresis where we consider the amino acids in the protein and whether they're acidic or basic, and whether the protein as a whole is positively charged or negatively charged. And the way that this works is that on our gel, we're going to have a pH gradient. So in the middle of the gel, it's pH 7, at one end it's pH 1, at the other end it's pH like 13 or 14. And remember that amino acids have protons that can be given away, and some places where they can accept protons. In an intact protein, the amino group and the carboxylic acid group are going to be tied up in the amide or peptide bonds, but any side chain or R group that has like a special functional group that can give away a proton, so that's going to be aspartic acid, glutamic acid, or to accept a proton, so that's going to be our arginine, lysine, histidine, that's going to affect the charge of the protein. And that protein is then going to want to migrate based on its charge, because remember that proteins migrate based on their charge. So in our pH gradient, The negative end is our basic end, and the positive end is our acidic end. And what we're looking for is our protein to travel to its isoelectric point, which means the pH at which the protein is electrically neutral. Now, you're not going to be asked to find the isoelectric point of a whole entire protein that is hundreds of amino acids long. You may be asked to find the isoelectric point of a specific amino acid, which would be take the pKa's of the various places of protonation or deprotonation on the amino acid and take their average, and that gets you the isoelectric point. But you should know what isoelectric focusing is and when it would be pertinent to use it, aka when I want to find out my isoelectric point of the protein or how many amino acids can donate protons versus accept protons. And this figure shows us how it works. Another really important lab technique in biology is called spectrophotometry. And this one, if you have done a biology class with a lab component or even a chemistry class with a lab component, you've probably done it. The way it works is that we put our sample in a little vial called a cuvette that's got clear edges, and we shine a light through it, and the machine lets us pick what kind of light that we shine. And that light of a specific wavelength is going to travel through our sample, and whenever there's a solute in there that can absorb that light of that wavelength, that light is going to kind of get lost. Now there's a detector on the other end of the cuvette that's going to tell us how much light did we get back at the end. So absorption is how much light did we lose? How much light was there in the beginning that we did not get back at the end? And absorption is actually directly proportional to concentration. That is, we can make a graph that says if we have absorption, this is equal to some constant times the concentration. And often this is expressed as the extinction coefficient times the concentration. And sometimes you'll see the path length, but the path length, or L, in these scenarios is often designed to be 1 centimeter so that we don't have to take it into account. So you'll often just have absorbance is equal to some coefficient times the concentration. And what we can do with this is we can make a calibration curve where we take different samples of some known concentrations and we take their absorbances and then we make a line. And the slope of that line, if we graph absorbance on the y and concentration on the x, is going to be our coefficient. And then we can go and apply that line to unknown solutions and use the coefficient and the absorbance to find the concentration. One important application of this is called the Bradford assay. The Bradford assay is a way to measure the general protein content of a solution. It's not going to find us a specific protein. It's going to tell us how much protein, any protein, is there in this solution. And the way that we do it is first, we are going to make a calibration curve of known concentrations of protein. Then we are going to take our unknown sample and we are going to treat it with a dye that binds protein. And when that dye binds protein, it's going to absorb at a specific wavelength, and we're going to put it through the spectrophotometer, and we can find that way the concentration of protein in our solution. So you might see that in a lab or in a passage sometimes. And then another technique that involves light, but we're getting here more into physics than chemistry, is called X-ray crystallography. And if you ever heard the story about how the structure of DNA was discovered, there was this lab and this scientist called Rosalind Franklin, and she did X-ray crystallography on DNA. And the way that this worked was she would shine an X-ray toward the DNA crystal. So the DNA was put in a crystal, and the way that the light filtered through the crystal was called diffraction, and that would create a certain pattern on the receiving end of the X-ray crystallograph. And from this, you could deduce the structure of whatever the crystal was made of. So Rosalind Franklin took X-ray crystallography photographs of the DNA structure, and then these two guys named Watson and Crick came along, and they looked at the structures and they said, this is what we think it is. And Rosalind Franklin for a long time didn't get a lot of credit, and now, thankfully, she does. But the point of this is that we can use X-ray crystallography in order to determine the three-dimensional structures of various biological molecules. For example, this is often used with proteins to determine their secondary and tertiary structure. And it's based on the physics concept of diffraction, or the fact that light passing through little tiny openings is going to create specific patterns. So now we come to restriction digestion. Restriction enzymes are super cool. So they're a defense mechanism developed by bacteria in order to avoid bacteriophage infection. Bacteriophages are viruses that infect bacteria, and bacteria avoid their infection because phages work by injecting their DNA into the bacterium. Now, bacteria are smart, and they've come up with enzymes called restriction enzymes that can cut phage DNA by detecting certain sequences, and these sequences happen to be what we call palindromic. Now, in English, a palindrome is something that's the same forwards as backwards, like Bob or Anna. But in biology, a palindromic sequence is a little bit more complicated. So it has to be the same 5' to 3' in each direction. And remember that DNA runs anti-parallel, so 5' here and 3' here means 3' there and 5' there. So if I wanted to have a palindromic sequence, I could have something like A, T, T, T. A, T, T, T. So if you read 5' to 3' this way, and 5' to 3' this way, those two readings are the same. So it's not something that's the same forwards and backwards necessarily, but it's the same if you go 5' to 3' on each strand. Restriction digestion usually happens with palindromic sequences of like 4, 6, or 8 bases, and it can produce blunt ends or sticky ends. Blunt ends are kind of what they sound like. It's like this, where you just cut in a blunt straight line, and you create two ends that are not jagged. Sticky ends are where you're going to create two pieces, and they have little overhangs, and notice that the overhangs will base pair with each other. So this is restriction digestion. Now, every organism's DNA, just by chance, is going to have some palindromic sequences, and we call these restriction fragment length polymorphisms. A polymorphism is just a fancy word for this is a mutation, but it's not necessarily harmful. So a polymorphism is kind of like a mutation, but it doesn't show up in deleterious ways. So we can actually use these to tell apart different DNA sequences because we can cut them up with restriction enzymes, and we can do an assay that detects how big these fragments are. And you guessed it, it's gel electrophoresis. So actually I want to skip ahead a few slides, and we can look at southern blotting, which I thought I had somewhere in here, but I guess I just have a slide that's gel electrophoresis of DNA. So in southern blotting, we take restriction enzymes, we cut up DNA, and we see the differences between different DNAs based on the fragment lengths that they create when they are digested with a specific restriction enzyme. So that's southern blotting. And we can also look at PCR. PCR is polymerase chain reaction. This is our way of amplifying DNA into many, many copies over and over again, but without the use of a cell. In PCR, we're going to use heat to speed up the process of DNA replication. We make use of specific DNA polymerases from organisms that are what we call extremophiles, which means that they can live in crazy environments and still be okay. So the enzyme, the polymerase that works in PCR, is meant to work at really high temperatures. That's how the organism it came from lived, in like hot springs. So when we do PCR, we're going to heat up our DNA, which causes it to separate. We're going to have primers, and these primers are little pieces of DNA that are going to anneal or stick to our parent strands. And then we're going to have our DNA polymerase go along and replicate our DNA. We can also have ligase in our mixture to put together the primers and the polymerase created DNA fragments. And then we do this many times over again, and we get many copies of our DNA that we can use in some experiment. For example, we can do something called transformation, where we put the DNA via like various chemical treatments into bacterial cells. So here's another figure of PCR. And one thing I should note here is that when we make DNA in the lab via PCR and try to insert it into cells, we often use it in the form of a plasmid. Plasmids in nature are little circular pieces of DNA in bacteria. But wait, bacteria always have circular DNA, right? Well, a plasmid is a little bit different because it's not the core genomic DNA in a bacterium. The plasmid has like what we like to call accessory genes. I think that's just my informal word for them, but basically it's things that the bacterium doesn't need to survive. So for example, it's not going to be enzymes that do glycolysis. What it might be though is, for example, antibiotic resistance genes might come on a plasmid, or the genes to be able to do conjugation, which is basically like bacteria sex, where they exchange DNA. So accessory genes or unnecessary genes are going to go on plasmids. And we can transform plasmids, like I said, transformation is like inserting DNA into a foreign cell that it didn't come from, and we can have that cell express that DNA. So for example, we can have the insulin gene put in on a plasmid, replicate it with PCR many times over, and then put it in a bacterium that can overexpress the insulin protein for our use as humans. So we looked at gel electrophoresis of DNA, we did that. We did bacterial amplification of DNA, so we can amplify DNA, make many copies of it through PCR, transform it into bacterial cells. These bacterial cells can make even more copies of our DNA. And if we want to isolate DNA from a bacterial culture, we can do something called mini-prep or maxi-prep, depending on how many bacteria we have. Okay, I've got to go fast because my time is going to run out on this app, but we have only a couple methods to go through left. This one is called Sanger sequencing. It's a type of DNA sequencing, and basically the way it works is that we're going to have multiple test tubes with different dideoxynucleotides. So what the heck is a dideoxynucleotide? Remember that in DNA nucleotides, we have a deoxyribose sugar. A dideoxynucleotide is a nucleotide that has another OH group missing on the sugar. So the DNA polymerase can add it on, but it can't add anything on after that because it doesn't recognize the sugar enough. So if we do this enough times to enough DNAs and enough different combinations, we can use these dideoxynucleotides to stop our reaction, our DNA replication, at every possible point. And then when we analyze this, we have DNA fragments of all different lengths, and we can make each dideoxynucleotide, A, G, C, T, fluoresce with a different signal so that we get our sequence. Now there are many second and third generation sequencing methods, but I'm not going to go over them here. Another thing we can quantify is the RNA, because remember that every cell has the same DNA, but what if I want to know not just what's there in DNA, but what's being actively expressed in the cell as RNA and as protein? Well, for one, we can use fluorescence in situ hybridization, or FISH. In this method, we're going to have a microarray of many nucleotide sequences that we want to test for. And these are complementary sequences to mRNAs we could potentially have in the cell. So if we isolate the mRNA in the cell and we expose it to this microarray, then we can see, okay, what's going to base pair, and when things do base pair, we're going to release a fluorescent signal. And then we can see what genes are actively turned on in our cell. Another method we can use is called RT-PCR, or reverse transcriptase PCR. In this method, we are going to take RNA that we've isolated from our cell and treat it with reverse transcriptase, which is an enzyme from retroviruses that basically takes RNA and makes it into DNA, so the reverse of transcription. Once we do this, we can treat it like DNA, do a PCR, and quantify exactly how much of it do we have, maybe compared to some control gene where we know how much of it is expressed. This is important, again, to see not just what's in our cell, but what genes are turned on and making RNA and making protein in our cell, which is super important. Well, I'm about out of time here, but I really hope that this helped you understand some research methods a little bit better.